Hello and welcome back, future scientist. I'm Mr. K with Mississippi Public Broadcasting, and I have another lesson today talking about active transport. How does the cell get things in and out of itself when it's trying to do these types of transport? So today we're really focusing on these two in particular. It's called vesicle and protein channel transport. These are both types of active transport, so these are going to require a little bit of energy in the form of ATP in order to accomplish them. So let's go ahead and hop into it and see what we got going on today. So the first thing I wanted to talk about, this is a type of vesicle transport. It's called endocytosis. Now I introduced this in the last unit, but I wanted to get into a little bit more detail. Now these pictures show a great example of what's going on here. Okay, in endocytosis, we're trying to bring something into the cell, and this is usually in a large quantity. So this is why we're going to use this method instead of diffusion or some sort of facilitated diffusion. Okay, so what's going on here is if you look at that picture, we're going to have all the molecules or whatever we're trying to bring into the cell around the cell. What's going to happen is the cell's going to kind of pinch in on itself and make a little pouch. You see that little circle there that has all those molecules inside of it? So that little circle is called a vesicle. You might remember back from way earlier in this class that a vesicle is an organelle, okay? It's how the cell moves things around inside of itself, okay? So when we have these molecules that we need, the cell's going to pinch off a little bit, make that little pouch, it makes that vesicle, and then it can bring it into the cell. And then we can use those molecules for whatever we need to. They might be food, they might be some sort of water or sugar. And so we're going to bring them into the cell so that the cell can use them, okay? So this is called endocytosis, and to me, this just kind of sounds like the first part of that word sounds like indoors, right? So this is how we bring something into the cell. So this is going to be the opposite from the other one that we'll talk about in just a little bit. So let's look into some more detail on endocytosis. We just saw a picture of it, but I kind of wanted to describe it this way as well. Okay, so in endocytosis, the cell's going to ingest external fluids, macromolecules, and large particles. This is stuff that would not fit through the cell membrane otherwise. What happens is those external materials are enclosed by part of the cell, and it's going to form a pouch, which was that little circle that we saw in the last picture. The pouch pinches off the cell membrane and becomes a membrane-bound organelle called a vesicle. Okay? Vesicles then can fuse with lysosomes. The lysosomes have digestive contents, and so that's how the cell would be able to break down those macromolecules and start to digest them. Okay? There's two types of endocytosis, and it really just depends on what you're bringing in. So those two different forms of endocytosis, we have phagocytosis. Phagocytosis means cell-devouring activity. And so what's going on here is this is how we bring in large particles that are solid. So phagocytosis is solid materials. Compare that to the other type of endocytosis called penocytosis. Penocytosis is how we move liquids into the cell. Okay, so again, this is endocytosis. There's two types, phago and peno, and it just depends on what type of material you're bringing into the cell. If it's a solid, we call it phagocytosis. If it's a liquid, we call it penocytosis. Great. So let's look at those in a little bit more detail. Here are some pictures to go along with it. Again, these are the two types of endocytosis. We have phagocytosis. This is when we're bringing in large solid particles. Penocytosis is when we're engulfing liquid particles. And this is just a picture that kind of shows how that works. So we have some sort of macromolecule or sugar or something outside of the cell that needs to come inside of the cell. The cell is going to approach it. It's going to start to surround it. It's going to kind of pinch off a little bit. That's called that vesicle that I mentioned earlier. And then the vesicle is going to move it into the cell so that we can use it. Okay? So this is all type of active transport. You can imagine that this is taking a lot of energy to have the cell move up to it, surround it, and then bring it into itself. Okay? So we are using energy in the form of ATP to make this happen. Okay, so that's all endocytosis. I wanted to talk about something a little bit different. This is called exocytosis. And exocytosis, we're having stuff leave the cell. So this might be waste that we're trying to get rid of. What's going on here is that the vesicle, that little pouch in the cytoplasm, is going to fuse with the cell membrane, and then it can release the contents to the external environments. This is how we release large molecules such as proteins that are packaged in the Golgi apparatus. So what would go on in this picture here, that vesicle would move in closer to the plasma membrane, it would fuse with it, and then it would release those particles to the external environment. So it's kind of like endocytosis in the opposite order. Instead of bringing it in, we're pushing it out. But it's kind of the same procedure where we're using a vesicle and it's pinching off, and then we're going to exit the cell. To me, exocytosis kind of sounds like the word exit, so this is how we're pushing things out of the cell. Okay, here's kind of another picture that kind of goes along with it. So remember that this Golgi apparatus, this picture down here, this is the Golgi apparatus. Okay, it's in charge of packaging and processing proteins, and we're also using it to transport things around. So what happens is when we're trying to get rid of these proteins off the Golgi apparatus, it makes these little vesicles. You see these little vesicles here? It's just kind of a little pouch that has the proteins in it. They kind of float over here to the cell membrane. They're going to fuse with the cell membrane, and then they can release those contents to the outside of the cell. So this could be some sort of waste or something that we need to get rid of. Okay? Great, so here's just a couple of different pictures to go along with it. This is endocytosis and exocytosis. What we really want to know is which direction these are going in. So in endocytosis, are we bringing things in or are we pushing things out? That's right, in endocytosis, we're going to bring things in. It kind of sounds like indoors, right? Exocytosis is the opposite. We're exiting the cell. Things are getting pushed out. So in this picture, you can kind of see what's going on. We want these molecules in our cell. We want these molecules in the cell. We make a vesicle. It fuses with the cell membrane. This is over on the right-hand side of that picture. We kind of pinch around it. We make a vesicle, and then we can bring it into the cell. So this is it. Fusing with the cell membrane. This is the vesicle. Then if this was some sort of macromolecule, maybe we want to digest this, it would fuse with a lysosome. Lysosomes have digestive enzymes, and so now when we get these two together, we're able to break those macromolecules down and use them for the cell. Okay, well, now that we've kind of started to digest them, we're going to have some waste, and we want that waste to be removed. So now what we're going to do is we take that vesicle. Again, this is just kind of a little pouch inside of the cell membrane. We're going to kind of push it out here towards the cell membrane. It'll do the same thing. It'll kind of start to fuse with it like this, and then we can release those wastes to the external environment and get them out of the cell. Great job. So here's the last little thing I wanted to talk about today. Again, this whole thing that we're talking about today is active transport. So the endo and exocytosis, that's vesicle transport. This is a little different. This is also a form of active transport, but instead of using vesicles, we're using protein pumps. So what's happening here is that small molecules and ions can be carried across the cell membrane by a protein in the membrane that kind of acts like a pump. What I really wanted to direct your attention to is look at that little explosion-looking part of this picture that says ATP. Now remember, ATP is the form of energy that your cells use, okay? It's adenine triphosphate. It's a molecule. It has 3 phosphates on it. If you break one of those phosphates off, it's going to release a little bit of energy. That's how, when we want to use these protein pumps, the protein pumps is going to change shape. And when it changes shapes, it's like opening the door, and so it can let things in and out of it. So over here, the door is open. The molecule can move through it, and then when we want to close it back, we use that ATP. The protein's going to change shape, and now the door's not open anymore. So this is another way that we can use active transport. We can use energy to move things up the concentration gradient and get things into the cell that we really need. So y'all, thanks so much for hanging in there with me today. Your teacher has some other assignments. Make sure to give that your best effort, and I hope everybody has a great day.